So we are continuing on the other structures of DNA that are possible. So some of those would look like this one, a triplex DNA, where you have 3 strands of DNA. So this is one strand, and another strand, wherein there is base pairing here, and also another base pair that is happening here with another strand. So and then you can also have a tetraplex wherein 4 DNA strands are interacting with each other via their nitrogenous bases. So basically, what this is just showing is that we don't have to confine ourselves into just 2 strands of DNA, although that is the most common and predominant form of DNA in nature. But the point of this slide is just to show you that other forms are also existing as well. So we can also have an HDNA, for example, here, which usually happens if one strand, for example, this green strand here, has a series of pyrimidines that are going, um, that are in sequence with each other. So because of this one strand kind of like has a shorter, it's less bulky than the other strand, and so the other strand, the black one, has a series of purines that are with each other, so they tend to be bulkier. So that's why this space here kind of happens. So this is an HDNA. Another DNA form, or nucleic acid form in general, that we can observe is a hairpin loop. So in a hairpin loop, you usually have a single strand nucleic acid. So this can happen whether you have a DNA or an RNA, wherein they will have sequences that are complementary to each other. And because these sequences are complementary to each other, your entire loop kind of folds on itself. So like you have a single-stranded nucleic acid, and this part here can be complementary to this part such that they form a loop structure. So this part here, where they have complementary strands, this is the stem part, whereas this part here is the loop part. So sometimes a hairpin loop is also called a stem and loop structure. So let's put our focus on RNA now. So there are 3 main types of RNA found in the cell. We have the mRNA, which is the messenger RNA. We have the rRNA, which is the ribosomal RNA, and we have tRNA, which is the transfer RNA. So basically, the messenger RNA is the one that carries information from the DNA towards the outer portion of the cell. The rRNA is a crucial component of the ribosome, which is the protein factory of the cell, whereas the tRNA, which is the transfer RNA, plays an important role in translation. So let's go through them one by one. So the messenger RNA, the reason why it's called a messenger is because the blue molecule here is actually your DNA. Now the DNA is found inside the nucleus, and it cannot get out of the nucleus. So whatever it is encoding has to be sent out using the RNA. So this is the messenger RNA, and it's single-stranded, and it is largely based on the sequence of your DNA, and it is this mRNA that goes out of the cell and goes out of the cell to go to your ribosome, so that whatever message it contains can be made into a protein. So its role is basically to communicate whatever message the DNA has inside the nucleus into outside the nucleus, where the ribosomes can read it. Okay, so that's the messenger RNA. Next, we have the transfer RNA, also known as the tRNA. So if you will find here, a tRNA it's actually single-stranded, but it folds on itself and it forms a cruciform. So this is called a cruciform because it looks like a cross. So if you will find it has like several stem and loop structures here, at least 3. So the transfer RNA is important because in this part here, this part is actually the one that complements the mRNA, so that the transfer RNA can read the message encoded by the mRNA, which is also the message that came from the DNA. And so by reading that, and in its other end, it contains an amino acid. So now, through the tRNA, we are able to translate whatever message the mRNA has into a protein component. So this actually here, the cruciform, shows the secondary structure of the tRNA, but it also folds on itself such that you have the tertiary structure, and the tertiary structure looks something like this. It's kind of like a reverse L form. So this is the part here that interacts with the messenger RNA, and this is the part here where the amino acid binds to. So next we look into the ribosomal RNA. So the ribosomal RNA is a crucial component of your ribosomes. So your ribosome is actually composed of an rRNA or a ribosomal RNA and some protein components, or the protein here actually serves as a scaffold. So it's kind of like a structure that helps to set up your RNA, because it's actually the ribosomal RNA that are needed to catalyze the formation of proteins. So it's not actually the proteins that are the important part of ribosomal RNA for its protein making mechanisms. It's actually the ribosomal RNA. Now, the ribosome has 2 subunits. It has a large subunit and a small subunit. So for prokaryotes, the large subunit is called a 50S subunit, whereas a small subunit is called the 30S subunit. In the small subunit, you find the 16S ribosomal RNA, whereas in the large subunit, we find the 23S rRNA and the 5S rRNA. So basically, this term 23S and 5S, they are the conditions at which these RNA were isolated, and now they are used to name the rRNA. So, kind of like they're lazy about naming stuff, so they just say, okay, I isolated this at 23S conditions, so I might as well name the rRNA as 23S rRNA. So it's something like that. So this is for the prokaryotes. Whereas for the eukaryotes, the small subunit of the ribosome is called the 40S, whereas the large subunit is called the 60S subunit. Now the 60S subunit, in there you will find the 28S and the 5.8S rRNA, as well as a 5S rRNA. Whereas in the small subunit, we find the 18S subunit. So these rRNA components plus the protein components form each of these individual subunits. And these individual subunits have to interact together to form an entire ribosome. So the entire ribosome for eukaryotes is called the ATS, whereas the prokaryotic counterpart is called the 70S ribosome. Now take note that this 70S here is not something that is a sum of the 50S and the 30S subunit. Remember that the 50S and the 30S are just indicative of the conditions with which these subunits or molecules were isolated, and hence they were the ones used for naming them. So they're not additive in nature. You don't say 50S plus 30S equals 80S, okay? 70S is the condition at which this subunit or this entire ribosome was isolated. So it's not a product of the addition of a 50S and a 30S, okay? So these are experimentally derived conditions. And consequently, they were the ones used to name the assembled components as well as the individual subunits. There are also other types of RNAs that are worth looking into. An example of that is the small nuclear RNA, or also known as the snRNA. Now, the snRNA is important for post-transcriptional modification. So because that's where it exerts most of its action. And your snRNA actually interacts with a protein such that they form a SNRP. So this is also called a small nuclear ribonucleoprotein. And this snRNA plus the SNRP, or specifically this snRNA, acts as a catalyst. Okay, so because it acts as a catalyst, we can also call this a ribozyme. So it would be wrong to say that only proteins have catalytic functions. Some nucleic acids like snRNA and rRNA also acts as catalysts. And so if you will find here, you have an mRNA, and this is further processed by the snRNA. So the U2 is actually a SNRP. So that means it has an snRNA component with a protein component, where once again the protein just serves as a scaffold. So it's basically a structure just to support the snRNA, so that you can make the snRNA environment more accessible for the substrates. So another RNA type I'd like to focus on, other types. So it would be the siRNA, or the small interfering RNA, as well as the miRNA, or also known as the microRNA. Now, both of these RNAs function in RNA interference, meaning they are specifically produced or expressed so that they can cut up some messenger RNA that either you don't want to use, or that are unnecessary, or that are sourced from viruses. So basically, the both of them will have similar functions in that they degrade other RNA, thus interfering with the RNA. Although there will be specific mechanisms as to how they are produced, which we will not be expounding on. It will be sufficient for you to understand that there are different types of RNA, and each with different purposes. However, we are just focusing our discussion on these 5 RNAs because they are the major types. Now, take note, I'm not saying that they are the only types of RNA. I'm just saying that they are the 5 major ones. And this ends our discussion about DNA and RNA structure.